Alrighty, so this would be your lecture midterm review. So we're going to cover chapters 1, 3, 4, 5, 6, 7, 8, 11, 12, and 13. Right? It'll be 50 questions or so. You have 115 points that you can earn on this midterm for the lecture portion of micro, and you'll be given 100 minutes to complete it, right? So when we're looking at chapter 1, right? Remember, it was Antony van Leeuwenhoek, right? He was the first person who saw microorganisms under a microscope and actually recorded it, right? Began using and making and using simple microscopes, often made a new microscope for each specimen, right? But he examined water and visualized tiny animals, right? Fungi and algae, and he called them animalcules, okay? And by the end of the 19th century, these organisms were then called microorganisms. But remember, he was the first person who saw the first living organisms. Okay? Now, microbes can be classified, right? We have bacteria and archaea. These are going to be unicellular, and they're going to lack a nuclei. That's what makes them a prokaryote, right? They're going to be much smaller than eukaryotes, found everywhere sufficiently where there's moisture, right? And some in even isolated extreme environments, right? But bacterial cell walls, remember that they're going to contain peptidoglycan in their cell wall, right? Not all of them, but most of them will. And archaeal cells, right, or archaea, right, they're not going to have peptidoglycan in their cell wall, right? They're going to have a different polymer, okay? But remember, prokaryotes, unicellular, they lack a nuclei, okay? Examples being bacteria and archaea. Fungi is a microorganism, but it's a eukaryote, right? So it has membrane-bound nucleus, right? And these fungi, they're going to differ from plants because they're going to obtain their food from other organisms, but they're going to differ from animals because they're going to possess a cell wall. Now, these fungi, they are not photosynthetic. Okay? They're not photosynthetic. So you're going to have two forms of fungi. You're going to have modes, which are multicellular. They're going to grow in long filaments. And then you have yeast, which are unicellular, right? So remember, fungi, not photosynthetic, right? And again, remember these modes are multicellular and they're going to grow in these long filaments, okay? Protozoa are these single-celled eukaryotes. Again, single-celled eukaryotes. They're generally motile. They're going to live freely in water. Some even live in animal hosts, but they are not photosynthetic. They're not photosynthetic, and most are going to be capable of locomotion, right? So they can move, right? Via it's a pseudopod, cilia, or a flagella. Okay? Remember these different scientists in the golden age of microbiology, right? Wells, right? Really promoted hand washing, right? Specifically preventing purple fever, right? Joseph Lister, remember Dr. Lister, right? Developed those antiseptic techniques. He pioneered the use of chemicals that will reduce infections during surgery. Okay, remember Wells, purple fever, Joseph Lister, right? Reducing infections during surgery. Florence Nightingale, right? Developed those antiseptic techniques in nursing and setting that standard of hygiene, right? Remember Paul Ehrlich, we talked about him before. Paul Ehrlich's magic bullets, right? Developed that field of chemotherapy, looked at chemicals to find a magic bullet that would destroy pathogens while remaining non-toxic to humans. Okay, so remember these scientists. Okay? What causes the disease, right? So remember, one of the first steps to identifying a bacterium, we use the Gram staining technique. The Gram staining technique is going to be named after Hans Christian Gram, right? Again, this is going to be the most widely used staining technique. Gram staining is the most widely used staining technique. And again, it's going to be one of the first steps to identify a bacterium. Remember these fields of microbiology, specifically the ones I highlighted, right? Serology, right? It's going to be using antibodies in blood serum, particularly as an indicator in infections. We have chemotherapy, like I just mentioned, the development and use of drugs to treat infectious diseases. And again, chemotherapy is where we use these chemical agents to harm or kill a microbe. Okay. Then, of course, we have bioremediation. This is one of those applied environmental microbiology fields where we use microbes to remove pollutants. Basically, this bioremediation helps to restore the environment. Okay. Then we have recombinant DNA technology, or like genetic engineering, right? where we thoroughly use this modern discipline, right, as a new way to treat infectious diseases, right? And of course, an area of genetics has been a major driver in modern microbiology. Okay, so remember this. Okay. Remember Koch's postulates too. Koch's postulates can be used to determine the cause of infectious diseases, right? Number one, that suspected causative agent must be found in every case of the disease and must be absent from healthy hosts. Then, so this is an order. Remember the order of it as well, right? The suspected causative agent must be found in every case of the disease and must be absent from healthy hosts. That agent must be isolated and grown outside of a host. And when that agent is introduced into a healthy host, a susceptible healthy host, that host must get the disease. And that same agent must then be found in the disease experiment host, right? So remember, again, Koch's postulates can be used to determine the cause of infectious diseases. We have that taxonomic classification system, right, from Carolus Linnaeus, right? And so this system basically, again, classified organisms based on similar or common characteristics. Okay, we were to group organisms that successfully interbreed into categories called species. And we use this term nomenclature as how we name organisms, right? This binomial nomenclature is the system of how we name organisms. When we're looking at different organisms, remember, viruses, we can't use a compound light microscope, right? Like how we would for bacteria, for example, right? Viruses are so much smaller, right? On that nanometer, right? You see NM, that's nanometer scale, right? Bacteria is on a micrometer scale. So you can't use a compound light microscope to view a virus, right? Because they're just so small. Remember Louis Pasteur, right? The father of modern day microbiology, right? Or the father of biology, right? The greatest biological scientist of all time, some is regarded as, okay? So remember that. Remember that theory of spontaneous generation. Many scientists believed in spontaneous generation. Then Aristotle believed in it too. A lot of people then really believed in it. They took a part of that, right? And remember the different scientists, specifically remember Needham, right? Needham, he was a scientist in favor of spontaneous generation, right? However, he failed to heat his vial sufficiently to kill a microbe, right? Or he didn't simply seal that vial enough. But it was Lazaro Spallanzani, right? So remember Lazaro Spallanzani, right? This person provided evidence to disprove spontaneous generation. Okay, so remember Needham was in favor of spontaneous generation, but Spallanzani disproved spontaneous generation. Okay. What causes fermentation? So scientists believe microorganisms cause fermentation, but it was Edward Buckner, right? He showed that it was actually enzymes that promoted the chemical reactions to undergo fermentation, right? So it's enzymes that allows for fermentation, okay? And again, it was Edward Buckner, his work began the field of biochemistry. Okay. We're looking at chapter 3, cell structure and function, right? Remember, these are the characteristics of life, right? An organism must be able to reproduce. It must be able to metabolize. It must be responsive to its environment, and it must be able to grow, right? Remember, these are going to be the characteristics of life for a living organism, right? So for something to be living, it must undergo all of these, right? So for example, a virus is not living because it can't undergo its own metabolism. It can't reproduce by itself, right? It needs to be inside of a host to do that, okay? Remember the external structures of bacterial cells, right? Such as fimbriae and pili, right? Fimbriae are going to be these sticky, bristle-like projections. Again, these are sticky, bristle-like projections. They're going to be used by bacteria to adhere to one another or to their environment. And these fimbriae are going to be shorter than flagella, but this fimbriae is going to serve an important function in biofilm formation, right? So remember fimbriae, sticky, bristle-like projection, right? And they are important in biofilm formation. Cilia are short hair-like structures. Cilia are only found in eukaryotes. The cilia is used for movement. Okay, they're going to be shorter and more numerous than flagella, but they're only going to be found in eukaryotes, and they're going to be used for movement. When we're looking at bacterial cell walls, right? So gram-negative cell walls have a thin layer of peptidoglycan, but remember that only in gram-negative will we find this lipid A portion. The lipid A portion is only going to be in gram-negative, okay? And this lipid A portion, because it is gram-negative, it may impede the treatment for disease, right? It may impede treatment for disease. And again, gram-negative are going to appear pink, right, following gram staining. Whereas gram-positive, they're going to appear purple, right, because they're going to have this relatively thick layer of peptidoglycan. Okay? They're also going to contain unique chemicals called teichoic acids and lipoteichoic acids. Right? So you'll find in gram-positive teichoic acids, lipoteichoic acids, and certain species will have mycolic acid. Okay? Remember that difference between gram-negative and gram-positive. Gram-negative have lipid A, right, in their outer membrane that will help them, resist against treatment. Gram-positive, teichoic, lipoteichoic, and mycolic acids, right? And I think it's really, really important for you to remember prokaryotic cell membranes, again, like I mentioned before, they don't have cholesterol, right? Our cells, like human cells or animal cells, we have cholesterol. Prokaryotic cell membranes do not have cholesterol. Okay? When we're looking at the cell membrane, right? So diffusion is the movement of a molecule down its concentration gradient from an area of high concentration to an area of low concentration. Diffusion allows for lipid-soluble molecules to cross over that cytoplasmic membrane, right? Osmosis is the movement of water. Gotta remember osmosis and how to understand osmosis. Yes, osmosis is the diffusion of water from high water concentration to low water concentration. So it's moving down its concentration gradient from high to low. But remember, water wants to move where there's stuff at, right? So osmosis is also the movement of water from low solute concentration to high solute concentration. Let's say it again. Remember, osmosis is the movement of water from low solute concentration to high solute concentration, whether that solute is salt or sugar, right? Water wants to move where there's a high amount of stuff at. Again, this goes over diffusion versus osmosis, right? Diffusion movement down their gradient, right? And again, you can have these lipid-soluble molecules that will move straight through a cell membrane because the cell membrane is lipid, right? Non-polar. Remember, when we look at the cytoplasm of bacteria, it's going to have cytosol. That's going to be the liquid portion of the cytoplasm, mostly water, of course. But we're also going to have these inclusions, right? These inclusions are going to be the sites of metabolite storage, okay? These inclusions are going to be the sites of metabolite storage. And remember, ribosomes, right? Just think about proteins. And so ribosomes specifically, they're going to be composed of several polypeptides and 3 RNA molecules and 2 subunits, right? So the 30S, right, or the 50S subunits, right? So remember that about ribosomes, okay? They're going to be made up of polypeptides and mRNA. Now, this is an important concept, right? These solutions, right? When the cell is in a certain solution, right? So if it's an isotonic solution, there's the same amount of stuff outside the cell versus inside the cell. So the cell is not going to change shape, right? Hypertonic is where there's more stuff. It's hyper, more than, right? Stuff in the solution. So remember, water wants to move where there's stuff at. So the water will move out of the cell into where there's stuff at. And then unfortunately, the cell will shrink up, crenate, and die. Remember hypotonic, right? This is where there's not a lot of stuff in the solution. It's hypo, it's little than, less than enough stuff in the solution. And there's more stuff in the cell. So water will rush into the cell and the cell bursts. Remember hypo for hippo. This cell is going to become big like a hippo and then it's going to burst, okay? Hypo. And also, so cells without a cell wall, right, such as these mycoplasmas, right, they're going to lack cell walls, so they require isotonic environments. So they require something like this, right, an isotonic environment. So remember, mycoplasmas require isotonic environments. Again, hypotonic solutions, that cell would become a hippo, enlarge, lyse, it'll burst. Okay? Remember that glycocalyx, and remember it can form like a slime layer, and that slime layer, just like a glycocalyx, remember it's like sugar, right? Sugar, carbohydrate, right? So remember the word. And I talk about it again, this glycocalyx, sorry, it is performs many different functions, right? So remember all these functions too. It's going to perform, responsible for cellular recognition and communication, protection against dehydration, strengthening the cell surface, anchoring the cell to each other. Remember, along with membrane, the glycocalyx, it plays a huge role in biofilm formation. Okay. Also, the glycocalyx will contribute to a bacteria's ability to cause disease. Okay? Remember these about the glycocalyx. Other non-membranous organelles that's inside the cell, we have centrioles. Remember centrioles. These are going to be non-membranous, okay? Non-membranous organelles, but they're going to be found only in eukaryotes. Not all, but only in eukaryotes. Now, and this helps with cell division or to move the DNA to the right place. Again, centrioles are non-membranous organelles found only in eukaryotes. A membranous organelle, right, so it has a membrane around it, such as the endoplasmic reticulum. The endoplasmic reticulum is basically a network arrangement of flattened hollow tubes continuous with that nuclear envelope, but it functions as a transport system. Okay, remember this about the endoplasmic reticulum. It functions as a transport system. There's two forms of the endoplasmic reticulum. There's the smooth endoplasmic reticulum, and write this down. Smooth is for lipid synthesis, to make lipids and steroids. The rough endoplasmic reticulum, it has the ribosomes on it. That's why it's rough. And if it has ribosomes on there, right, it's going to be important for protein synthesis, right? So again, smooth ER, lipids, rough ER, proteins. And again, it functions as a transport system. Okay. Now, these processes of endocytosis and exocytosis. Endocytosis is bringing substances in the cell that you can see right here. Circling with my mouse is bringing it into the cell, inside a vesicle, right? Endocytosis. Exocytosis is where it's leaving the cell. And so both of these processes are a means of transport by eukaryotes, right? So you can transport substances such as prokaryotes in it, for example. But again, both of these processes are a means of transport used by eukaryotic cells. When we're looking at chapter 4, right? Microscopy, staining, and classification, right? I told you before about those viruses. You can't see them under a light microscope because they're so small. Viruses are measured in nanometers. And if we look at nanometers, remember nanometers is right here. 1 times 10 to the negative ninth. Write that down. Nanometers, 1 times 10 to the negative ninth. I also want you to write down micro, which we're studying. Micro, like a micrometer, is 1/1,000,000th. If it's 1/1,000,000th, micro is 1 times 10 to the negative 6. 1 times 10 to the negative 6. So nano, because it's times 10 to the negative 9, it's much smaller than micro, okay? Again, nano times 10 to the negative 9, micro times 10 to the negative 6. And then I also want you to remember milli, like a millimeter, right? Notice how it's like 1/1000 is 1/1000th, and that's 10 to the negative 3, right? So remember those. Milli, 1/1000th. Micro, 1/1,000,000th of a meter. And then remember, nano is 10 to the negative 9. And this is what viruses are measured in nanometers. Okay. When we're looking at light microscopes, remember these magnifying lenses are going to be the ocular lens and the objective lens. Again, these are going to be those magnifying lenses. And sometimes we use that immersion oil. And that immersion oil is there to improve the resolution because it increases the numerical aperture and maintains a uniform light speed. Okay, so remember these about microscope. Okay. Also, remember that a phase contrast microscope is used to examine living organisms, okay, or specimens that would be damaged or altered by attaching them to a slide. But remember, you know, these phase contrast microscopes are used to examine living organisms. Okay. So we're looking at something, right, such as a simple stain. It gives us morphology, right? And morphology is the size. Morphology is simply the physical characteristics. What does it look like? Right? What is this morphology? What does it look like? Okay, remember that. Remember Carl's compared nucleotide sequences of RNA sequence or subunits, ribosomal RNA, that is what it is. And we're able to propose 3 domains as determined by the ribosomal nucleotide sequence. So we have those 3 domains being eukarya, bacteria, archaea. Understand and remember, viruses are not there. Viruses are not there because they lack ribosomal RNA. Simply, they lack ribosomal RNA, so they're not in a domain by themselves or in any domain. Okay, that's why they're not required. They're not described as living. 2, again, taxonomy consists of classification, and I mentioned earlier, nomenclature is that naming system. Nomenclature is that naming system. And so the placement of an organism into a domain, like I mentioned in the previous slide, is made on the basis of ribosomal RNA. Viruses are not included in this taxonomic scheme because they lack ribosomal RNA. Okay. Staining that I talked about earlier, right? If we use differential staining, right, that's using more than one dye. Simple staining is one dye. Differential staining is more than one dye, 2 or more dyes, right? So differential stains are used to distinguish between different cells, chemicals, or structures. Remember, gram staining, notice I was pink. Gram staining shows differences specifically in cell wall, right? It's either gram-positive cell wall or gram-negative cell wall. But again, gram staining shows differences in the cell wall, okay? So dyes, when we're looking at staining, dyes are used as stains. There's usually going to be salt. We have chromophore. This is going to be a colored portion of the dye. Acidic dyes, these are going to stain alkaline structures. These are going to be like those negative stains. And we have basic dyes, which will stain acidic structures. So again, acidic dyes stain alkaline structures. Basic dyes stain acidic structures. Okay. And again, these basic dyes, these are going to be more common since most cells are negatively charged anyway. Chapter 5, when we're looking at microbial metabolism, right? Remember the 2 classes of metabolic reactions: catabolic, breaking something down; anabolic, right? We're synthesizing larger molecules from small ones. So we're building up something. We're using small ones and we're building up to a larger molecule, right? That's anabolism, okay? Again, we're building up something. Now, enzymes are types of proteins. Tell you all the time, proteins do everything in the body, right? I don't want you to have to remember every type of protein, right? But remember certain ones: oxidoreductases, throwing and receiving of electrons; ligases, right? You're ligating something. You're putting something together, right? So that's anabolic. You're putting something together. You're growing something, right? That's anabolic reactions. Lyases, right? This one right here, that's breaking something down. Catabolic. So it's the opposite. All right. Remember those 2, right? Ligases versus lyases. Okay. Also understand these oxidation reduction reactions. If something accepts an electron, right? If it accepts an electron, it's reduced. Okay. If it loses, so this one right here lost an electron, it is oxidized. So if we remember, I'm going to write on the screen really quickly, right? Oil rig. Again, I'm not gonna write it out though, but oxidation is loss. This one is oxidized, so it lost its electron, right? And then we have reduction is gain. This one right here, it gained an electron, so it was reduced, okay? Remember that oxidation is lost, reduction is gain, okay? Well, gonna have to remove that. All right, clear all the drawings. There we go. Now, let me go back a second, sorry. Enzymes are types of proteins, right? Enzymes act on substrates. Remember that. Enzymes act on substrates. They only work in certain conditions, right? So if this enzyme is in the body and it's supposed to work in, 7.45 pH or 37 degrees Celsius, if you take it out of that range, it's not going to work anymore. Okay, so remember, it only works in certain conditions. Okay, notice how we have this enzyme right here. It will act on a substrate. And when we look at how it acts on the substrate from like number 1 to number 2, number 3, number 4, and 5, right? This enzyme is unchanged at the end. It's unchanged. So we can use this enzyme over and over and over again to do that same job. Okay. So remember, it's going to work on a substrate. It's going to be used over and over, right, to do the same job. It'll come back out of this chemical equation completely unchanged, ready to do it again, but it only works in certain conditions, right? In certain pH, certain temperature, right? Certain alkalinity, or pH salt, certain conditions, right? If you take it out of that condition, it's not going to work because it's a protein. Now, we have certain things that so this is an enzyme. So notice how we have the active site right here, and this is an allosteric site. An allosteric site is any site that's not the active site. So we can have something that binds there at that allosteric site, and it changes the shape of the protein. So it changes the shape, right? It distorts that active site. And so because it did that, it acts as an inhibitor, specifically an allosteric inhibitor, also called a non-competitive inhibitor. And then we also have some things, notice how the substrate can bind in there, but this inhibitor can bind there too. So because it's binding at the active site, it's competing for the active site. So that's an example of a competitive inhibitor. Okay, so we have an allosteric inhibitor that binds somewhere that's not the active site, and we have a competitive inhibitor that's competing at the active site. Okay. All enzymes are not always active. Sometimes we synthesize an enzyme and it's inactive, right? And we need to activate it. So we have these cofactors or these coenzymes to bind to an enzyme to activate it. Example I give you, a vitamin. That's why we need vitamins. Vitamins act as coenzymes. Vitamins act as cofactors. They bind to an enzyme and make it active again, okay? Or they make it active. Remember this chart right here, because you'll see it again. Remember that this part is glycolysis. This is citric acid cycle. This is electron transport chain. And if you don't have oxygen, this process right here is fermentation. Okay. Carbohydrate metabolism is commonly used to identify bacteria. So if I ask you, well, what is one process that's commonly used to identify bacteria? Carbohydrate metabolism. Can this bacteria metabolize a sugar, right? And what kind of sugar can it metabolize, right? So if it metabolizes the sugar, right, that's good. Okay? Glycolysis is the breakdown of glucose into forming pyruvic acid. Remember that. Glycolysis is the breakdown of glucose into forming pyruvic acid. If you have oxygen, you're able to make a lot of ATP. Because if you have oxygen, you'll go through cellular respiration, this process right here, right? Again, glucose is broken down to make pyruvic acid. Pyruvic acid then gets converted to acetyl-CoA. It's the acetyl-CoA that then gets siphoned into the citric acid cycle to make the NADH, the FADH2, and then FADH2, NADH. They have these electrons. They drop off their electrons, as you can see right here, to the electron transport chain. And it's from the electron transport chain that provides the energy for oxidative phosphorylation. Okay, that oxidative phosphorylation is where we're cranking that ATP synthase molecule, right? And we're phosphorylating ADP to make ATP. That's the process, okay? You also have to remember that in aerobic respiration, oxygen is the final electron acceptor. Oxygen is the final electron acceptor. In fermentation, where oxygen is not present, then we're going to use an organic molecule as the final electron acceptor. But remember, when oxygen is available in cellular respiration, oxygen is the final electron acceptor. Okay? Remember these. Okay? Remember this image. In chapter 6, we looked at microbial nutrition and growth. Microbial growth is basically the increase in population of microbes. So it's not their increase in size, but it's the increase in numbers, right? And that's going to be due to the reproduction of individual microbes. So we have these discrete colonies, which is just simply an aggregation of cells arising from a single parent cell. But remember biofilms. Biofilms are important because this is a complex collection of microbes living on a surface, right, in a community. Okay. And again, it's fimbriae and glycocalyxes, right? They play a huge role in biofilm formation. Okay, remember, biofilms is a collection of microbes living on a surface in a complex community, and the glycocalyx and fimbriae, right, these outside structures, they play a huge role in this biofilm formation. Remember the different oxygen requirements of organisms. If it requires oxygen, it is an obligate aerobe. If it requires oxygen, it's an obligate aerobe. Okay. And you're going to find those going to be at the top of something. It's going to be at the top of water, or it's going to be at the top of a test tube, as you can see right here. Okay. If oxygen kills the species, right, it's an obligate anaerobe. Okay? Does not require oxygen because oxygen will kill it. You're going to find that at the bottom of the sea, bottom of this test tube, for example. Okay. And then you have facultative and aerotolerant. These can grow with or without oxygen. Again, with or without oxygen. Obligate aerobe must have oxygen. Facultative anaerobe or aerotolerant anaerobe, they don't need it, right? But they can grow with it or they can grow without it. And again, obligate anaerobe, no oxygen. It kills them. Okay. Remember, we have, bacteria that will thrive in certain conditions, whether it's the oxygen, like I mentioned, whether it's the temperature, or whether it's even like pH, certain pH, right? So, and it's not on the slide, but I'm going to talk about it for a second though, but it's like, say for example, you have an acidophile, something that likes acid, right? So it's going to like something that's low on the pH, right? So it'll live in acidic environments, right? Or a halophile. Halophiles like salt, right? So remember that too. Okay? You have these thermophiles, right? They like hot temperatures, okay? And so these thermophiles or these hyperthermophiles at these higher temperatures, right? They are okay, right? They thrive in these conditions, but for some organisms, such as these mesophiles, these mesophiles right here, these are the ones that be getting us, right? Because our body temperature is 37 degrees Celsius, right? And this is where these mesophiles, we get affected by them. But at higher temperatures, right, than the max for that particular organism, if it's too hot, those hydrogen bonds will break. right, between the DNA, hydrogen bonds break. Proteins are denatured. The membranes become too fluid, right? So remember this, okay? Okay. Okay. Remember the steps in binary fission you will be asked, right? Remember number one, the DNA is going to be replicated. The cell is going to double in size. Sorry, DNA is replicated. The cell is going to double in size, and the size gets doubled in size. Then we're going to form this septum or this cross bridge, and then it's going to separate and form two offspring. Okay. Again, we replicate the DNA. The cell doubles in size, forms that septum, splits off to form its offspring. Okay, real simple, binary fission. And it's that binary fission process that happens during this log phase, really. And it takes really quick. And this is that, so let's look at this typical population of a growth curve, right? Now, remember, the growth of bacterial cultures is best described as logarithmic growth, okay? Especially because we see this log phase. So it has a lag phase. You will be asked about these different phases, though, for sure. It has a lag phase. The lag phase, notice how it's just lagging, right? Cells are there, right? They're metabolically active, but they're not dividing. So they're just getting used to this nutrients, right? They're not growing in numbers. They're still living though, right? But they're just, they're not dividing. They're just getting used to the nutrients. Then you have this exponential growth. That's this log phase. This is where the cells are growing and actively dividing. Okay. And then we have the stationary phase. So remember, the stationary phase is where you're going to have cell growth equal to cell death, right? So just amount is growing and it's equal to just amount is dying. Okay. And then the death or the decline phase is where they're dying more rapidly, basically. Okay. So remember those different phases. Okay. Also, I want you to remember the generation time, right? The generation time is the time it takes for the cells to double in number. This is going to be, the generation time is shortest, remember, is going to be shortest in the log phase. Okay, generation time is going to be shortest in the log phase. Okay, because again, they're growing logarithmically. They're growing really, really fast. Okay? Some might have a generation time of 18 minutes, like E. coli has a generation time of 18, 20 minutes. So it doubles. That binary fission doubles every 18 minutes logarithmically. Okay? Remember this, you'll see it. Sorry. One of the indirect methods to measure microbial growth would be turbidity. Okay? Turbidity, an indirect method to measure microbial growth. And that's just measuring the amount of light that can go through this tube. Okay? Chapter 7, microbial genetics. When we looked at the structure of prokaryotic genomes, when we're looking at prokaryotics, right, or prokaryotes, remember they're going to have a circular DNA molecule and it's going to be in the nucleoid region. They're going to have circular DNA in the nucleoid region. Okay, these prokaryotic cells are going to be haploid and a single copy of the DNA. Versus eukaryotes, right? Right here, eukaryotes, we're not going to have circular DNA, right? We're going to have linear DNA. And we're not going to have one chromosome per cell. We're going to have multiple, okay? We're going to have multiple, and they're going to be linear. Now, prokaryotes, like bacteria, right? They'll have these plasmids. Plasmids are going to be these small molecules of DNA that can replicate autonomously or independently, okay? I give you an example right here that's highlighted, right? These bacterial plasmids. These plasmids will carry genes for proteinaceous toxin. Basically, they'll kill bacteria of the same or similar species, right? For competition, right? So bacteria, they can produce these plasmids that will carry genes that will kill, right? The same bacteria or similar bacteria so they can reproduce, basically. Right? And again, remember that these plasmids, again, are small molecules of DNA that can replicate autonomously or independently. When we're looking at DNA replication, right? Replication is semi-conservative. Replication is semi-conservative. What does that mean? That means each daughter DNA molecule, right? Each daughter DNA molecule is going to be composed of one of the original strands and a new strand. So we have the original strand, this purple one, and we have a new strand, right? This blue one. And then again, that daughter cell will have one of the original strands, the purple one, and a new strand right here, right? Or if it's right here, an original strand from this daughter cell to this right here, to blue, and then a new strand. So that's semi-conservative DNA replication, right? Each daughter DNA molecule is going to be composed of one of the original strands and a new strand, okay? Also, remember this lagging strand. Just remember this image and how it looks. This lagging strand is discontinuous. Right? Notice how it's not continuous like that. It has this gap in it. It's discontinuous and it's much slower. Okay, lagging strand, discontinuous, much slower. Remember the central dogma. DNA is transcribed into RNA. RNA is then translated into protein. Okay? DNA to RNA, transcription. RNA to protein, translation. Okay? Remember the different types of RNA. Specifically, I want you to remember tRNA. This is what tRNA looks like. It's transfer RNA. The role or the job of transfer RNA is to deliver the appropriate amino acid to that growing polypeptide chain. Again, tRNA delivers the appropriate amino acid to the growing strand. When we're looking at gene function, right, especially these operons, an operon consists of a promoter and a series of genes. Again, an operon will consist of a promoter and a series of genes, right? And it's going to be controlled by an operator. It's going to be controlled by an operator. An operon is going to be consistent of a promoter and a series of genes, and it's going to be controlled by an operator. Remember mutations, right? They can be good or bad, right? But mutations is basically a change in the nucleotide-based sequence, right? They're pretty rare, right? They are almost always deleterious. Remember, a point mutation is where one base pair is affected, okay? So a silent mutation leads to no change in the protein sequence. It's silent, so nothing happens, really. Okay? Mutagens, remember this, right? Mutagens, so such as like radiation, for example, ultraviolet light can create these thymine dimers, okay? Ultraviolet light can create these dimers. And notice how you have this covalent linkage between two thymines. That's a thymine dimer. And that's what ultraviolet light does. That's what sunlight does, okay, to your DNA. And it creates these thymine dimers. Chapter 8, when we looked at recombinant DNA technology, remember biotechnology is the use of microorganisms to make practical products. The use of microorganisms to make practical products. I give you an example. Bacteria that produce insulin, right? Remember, we used to get insulin from pigs, but now put the gene in the bacteria. Bacteria can now produce this protein, insulin, right? Biotechnology, the use of microorganisms to make practical products, right? Use this microorganism, use this bacteria to make a practical product such as insulin. Mutagens, like I just mentioned, right? They can create changes in microbes' genome, right? And this can be beneficial, right? For us as a scientist to use this. We can create changes in a microbe's genome to change their phenotype, right? Remember restriction enzymes. Restriction enzymes are these bacterial enzymes that cut DNA molecules, right, into fragments. They cut DNA molecules, specifically at restriction sites, but they're going to cut this DNA into fragments, okay? You have sticky ends and blunt ends. Remember PCR, polymerase chain reaction. Polymerase chain reaction, right? We put our DNA, right, strands of DNA, in a machine called a thermocycler. Okay. We put our reactants, right, DNA, your nucleotides, things like that, your enzyme, your DNA polymerase enzyme, of course, in a thermocycler. So, gotta understand too. When we're using this thermocycler, is heat, all right? It's gonna go through cycles. Okay, just thinking about the name. We're heating up this DNA. And the reason why we're heating up this DNA is because this whole process, we're making multiple copies of the DNA. And in order to do that, we need to break it apart and make more copies. In order to break the DNA apart, remember, it's held together by hydrogen bonds. So it's the high temperature that's important for breaking the hydrogen bonds. Okay? It's the high temperature that's important for breaking the hydrogen bonds that you can see right here. Okay? But remember, in order to add the nucleotides, you need a DNA polymerase. So you can't just use a regular DNA polymerase that you'll see from like E. coli or something like that, right? You can't use a regular DNA polymerase. You have to use a specific heat-tolerant DNA polymerase enzyme, right? So we use Thermus aquaticus DNA polymerase. Remember that, okay? We use Thermus aquaticus DNA polymerase because it can withstand the high temperatures. Okay. And again, this whole process, this PCR, is there to amplify the DNA. We're going to put this DNA again in a thermocycler machine. Okay. Gel electrophoresis, you'll see these questions too, right? Gel electrophoresis is a process that separates molecules, whether it's DNA or proteins, for example, but it separates molecules based on their charge, their size, and their shape. Okay. So DNA is negatively charged. So you'll put your DNA in the wells up here on the negative side. Notice how it's negative side right here. Okay, it's the negative side. And when you turn the machine on, the DNA will migrate to the positive side. Okay. So again, that DNA, negatively charged DNA, will migrate to a positive end or the positive electrode. Okay. And so because we're separating it based on their charge and based on their size, right? The smaller fragments, it's able to migrate and get through this agarose gel. So the smaller fragments will migrate faster and farther. So for example, you'll have DNA down here, right? It might be 150, 200 base pairs, right, in length. But up here, right, up here where my mouse is at, obviously, you'll have DNA fragments that are larger, like 5,000 base pairs, right? Because it was just too big to get through down here, right? So bigger ones will be up here, smaller ones will go through. And again, gel electrophoresis separates molecules based on charge, size, and shape. The negative DNA will be attracted towards the positive end, and smaller fragments will migrate faster and farther than larger ones. Chapter 11, we're looking at characterizing and classifying prokaryotes. Remember the morphology of prokaryotes, right? And so, for example, cocci are spherical, bacillus are more rod shaped, all right? These pleomorphic bacteria, they're going to vary in size and in shape, right? They don't have a defined size or shape, so they will vary. Give you an example, mycoplasms, right? These are examples of pleomorphic bacteria, right? Because they don't have a cell wall, so they vary in shape. These spirochetes, right? These are flexible spiral-shaped bacteria. So flexible spiral-shaped bacteria. And then we have this manual, this Burgess Manual of Systemic Bacteriology. So it basically contains these classification schemes for prokaryotes, right? And it's this manual. Again, it contains these classification schemes for prokaryotes, and it's going to be based on ribosomal RNA sequences. Burgess Manual of Systemic Bacteriology is based on ribosomal RNA sequences. Okay. Endospores, okay, are produced by gram-positive bacteria only. Okay, I give you an example. Bacillus, remember these examples. Bacillus, Clostridium species, right? They produce endospores. And they're going to produce these endospores because there's a defensive strategy, right, against unfavorable conditions, right? So they'll produce these endospores when nutrients are scarce. When nutrients are scarce. Okay. But of course, these endospores, these are a concern to food processors, healthcare professionals, and governments because these are highly resistant to killing. Okay? It's really hard to kill these endospores. That's why we use an autoclave. High temperature, high pressure. Okay. Archaea, again, they don't have a peptidoglycan wall that's going to be made up like the bacteria, right? We can distinguish between bacteria and archaea by their cell wall composition, their cytoplasmic membrane, lipids, right, and their ribosomal RNA sequence. Okay, so remember that. Okay? And again, these archaea, they're in extreme environments, so where we are not, so they're not known to cause any disease, though. Okay. Methanogens are a type of archaea. These are going to be the largest group of archaea. Remember this. Methanogens are going to be the largest group of archaea. Okay, they're going to convert carbon dioxide, hydrogen gas, and organic acids to methane gas. Okay. Thermophiles, all right? Their DNA, RNA, cytoplasmic membranes, and proteins, right? They don't function below 45 degrees Fahrenheit. So they function in high temperatures, right? Remember the example of hyperthermophiles. Hyperthermophiles require temperatures over 80 degrees Celsius, right? And they're found in places like hot boiling springs, right? Two representative genera, right? The Pyrodictium and the Thermococcus bacteria or species. Sorry, I should say. Again, hyperthermophiles are going to, again, be found in places like hot boiling springs. Okay. Have deeply branching bacteria. And so these were bacteria believed to be similar to the earliest bacteria found. Okay. Again, these were believed to be some of the earliest bacteria found. Okay. They're going to live in habitats similar to those thought to exist earlier on early Earth, basically. Okay. They're going to be autotrophic. Mycobacterium, right? You should already know this. Mycobacterium, they're going to have these mycolic acids in their cell wall. And it's because of these mycolic acids that they can grow slowly. Mycolic acids and these mycobacterium, they can grow slowly. And remember, Mycobacterium tuberculosis. Remember that name, right? It causes tuberculosis. Don't get Mycobacterium mixed up with mycoplasma. Mycoplasma, right? They don't have a cell wall. Mycobacterium, they have mycolic acid in their cell wall that makes them grow slowly. And species of Mycobacterium, right, causes tuberculosis. Okay. Oh, sorry. Hold on. Chapter 12, when we're characterizing and classifying eukaryotes. So when we're looking at the reproduction of eukaryotes, they're going to be a little bit more complicated than prokaryotes, of course, because most eukaryotic DNA is going to be packaged in chromosomes in the nucleus. But when two gametes come together, it forms a zygote. When two gametes, right, two sex cells, right, egg and sperm, for example, come together, it forms a zygote. Okay. Algae, fungi, some protozoa, they reproduce both sexually and asexually. Okay, remember, gametes come together to form a zygote. Okay, remember the phases of mitosis. You will see these. Okay. You don't see it right here, but up here, right? Before prophase, we have interphase. In interphase, this is where DNA replication occurs. Okay? Let's say it again. DNA replication occurs in interphase. Okay? When you replicate your DNA, right, then you can undergo the process of mitosis, which starts with prophase. The nuclear envelope, so you got to remember these, we talked about it before. In prophase, the nuclear envelope deforms. The chromosomes start to condense. That's why you can see them as like these X's, basically. In metaphase, right? It's not typed, but remember, write it down. In metaphase, these chromosomes, they line up in the midline of the cell, in the metaphase plate. In anaphase, notice how the sister chromatids are being pulled apart to opposite poles of the cell, opposite sides of the cell. In telophase, they're already at opposite sides of the cell. And notice how you see these dotted lines is the nuclear envelope is reforming back around it. Telophase is the opposite of prophase, basically. Okay. And then in cytokinesis, right? So I'm going to scroll down or go to the next one. Cytokinesis, this is where we would actually have, if I can write it in for a second, we would have this cytokinesis right here, this cleavage furrow coming to separate the two cells. Okay, that's in cytokinesis. Now we have two new identical daughter cells. Okay. Can I clear it again? Okay, for a second. In cytokinesis, right? However, I just mentioned you separate it and you have two new daughter cells. And some organisms, such as like algae or fungi, they will postpone their cytokinesis, or they might not even have it at all, right? And so you might have a cell that has a lot of nuclei in it. And these cells that are multinucleated, they're called these cyanocytes, or these coenocytes, I'm sorry, right? These coenocytes, right? This is where we did not have cytokinesis. Okay? And again, cytokinesis is cytoplasmic division. The cytoplasm, like oh gosh, right here, if the cytoplasm didn't divide and it didn't split off, right, it would have multiple nuclei in the cell, like how you have here, one nuclei over here, one nuclei over here, right? And it can have multiple ones, right? Multiple nuclei in a cell is called these coenocytes. Okay. Now, this is the process of schizogony. So you'll see this, right? Schizogony. So some protozoa, or like Plasmodium, right, that causes malaria, they'll reproduce asexually via a special type of reproduction called schizogony. So this is where we have multiple mitosis happening, followed by a single cytokinetic event. So you have all this mitosis happening, you're creating all these nuclei, and then finally you have one cytokinesis event, right, that's creating these daughter cells, right? So this is an example of schizogony, right? Multiple mitosis followed by a single cytokinetic event. Protozoa are these diverse groups, right? These are going to be eukaryotes. They're unicellular, and they lack a cell wall. Again, they're eukaryotic, unicellular, they lack a cell wall, and they're going to be motile by means of cilia, flagella, or pseudopods. Okay. Fungi have cell walls, do not perform photosynthesis. Okay. Fungi, they're able to decompose dead organisms and recycle their nutrients, and they produce antibiotics and other drugs. Okay. Types of fungi, modes, right, and yeast, right? But modes are going to be composed of these long filaments called hyphae. You'll see that. Okay. Modes are going to be composed of these long filaments called hyphae. Okay. Lichens, right? These are basically a partnership between fungi and photosynthetic microbes. So you have fungi and cyanobacterium, or fungi and green algae, basically partner together to form these lichens. Okay, to form these lichens. And again, it's partnership between fungi and photosynthetic microbes, such as cyanobacterium or green algae. Chapter 13, right? It's the final chapter, right? That's going to be in the midterm. Went over characterizing and classifying viruses, viroids, and prions, right? Viruses, they're not living, right? Because they're not, we classify them as not living because they don't have ribosomal RNA, right? They don't have cytoplasmic membrane, cytosol, or organelles either, right? Again, they don't have a cytoplasm, right? Cytoplasmic membrane. They don't have a cytosol. They don't have organelles, right? They have a protein coat and they have a nucleic acid core. It's called a virion when it's outside the cell. Okay, remember that. It's called a virion when it's outside the cell. That protein coat, that capsid, is responsible for the shape of the virus. Again, this outermost layer, right, is going to provide protection and recognition site for the host cell. So that's why it's able to infect your host cell, because it provides protection and a recognition site for your host cells. Okay. And again, oh, remember, viruses that infect bacteria, these are called bacteriophages. Viruses that infect bacteria are called bacteriophages. Okay. Viruses can either be DNA or RNA, can't be both. So this is one way we can classify or categorize a virus based on its nucleic acids. We can classify or categorize a virus based on its nucleic acid. It could either be a DNA virus or an RNA virus. Can't be both. It would only have DNA or RNA in it. Okay. Now, a virus will infect a host cell. So some viruses will only infect your liver cell or will only infect your brain cell, right? Because it's due to the affinity of that viral surface protein for complementarity on your host cell surface. So it must be, specific to, they may be so specific that they only infect particular kinds of cells in a particular host. And again, that's due to the affinity of a viral surface protein for the complementarity on your host cell surface. I mentioned it again earlier, capsids, right? They provide protection for viral nucleic acid. This capsid protein is also a means of attachment to your host cell. Okay? And they're primarily responsible for the shape of a virion. The capsid, this right here, primarily responsible for the shape of this virus, okay? When we're looking at viral replication, it could be lytic or it could be lysogenic. The stages of lytic replication, it attach, it enters, synthesizes more, it assembles, and then it releases more viral particles, right? So remember the 5 stages of lytic replication: attachment, entry, synthesis, assembly, and release. Okay. This goes over attachment of the virion to the host cell, entry into the host cell, synthesizing new nucleic acid molecules of viral nucleic acid molecules, assembling into new virions within the cell, right? And then release, right? Release of these virions from the host cell to infect more. Lysogenic is where the infected host cell grows and reproduces normally for generations before it actually undergoes lysis, right? So the genetic information of the bacteriophage is amplified many, many times before that cell breaks apart. Okay, and this is lysogenic. Okay, many, many times before it actually breaks apart. We can culture viruses in a laboratory. Remember, viruses only grow inside of a host, so we can culture viruses via a cell culture, like a tissue culture, as you can see right here. Cell culture, tissue culture, or we can culture viruses in embryonic chicken eggs, okay? Again, inside a host cell. Viroids, so remember, viroids are pathogenic molecules. They only target plants. Viroids target plants, okay? Viroids target plants. And prions, I'll end with prions. Prions are these proteinaceous infectious agents. They lack nucleic acids. That's what's special about prions. They lack nucleic acids, again, because they are proteinaceous infectious agents. Okay, so that was your quiz or midterm lecture midterm review. Okay?